RNA interference is a biological process in which RNA molecules are involved in sequence-specific suppression of gene expression by double-stranded RNA, through translational or transcriptional repression. Historically, RNA was known by other names, including co-suppression, post-transcriptional gene silencing, and quelling. The detailed study of each of these seemingly different processes elucidated that the identity of these phenomena were all actually RNA. Andrew Fire and Craig C. Mello shared the 2006 Nobel Prize in Physiology or Medicine for their work on RNA and the nematode worm Caenorhabditis elegans, which they published in 1998. Since the discovery of RNA and its regulatory potentials, it has become evident that RNA has immense potential in suppression of desired genes. RNA is now known as precise, efficient, stable and better than antisense therapy for gene suppression. Antisense RNA produced intracellularly by an expression vector may be developed and find utility as novel therapeutic agents. Two types of small ribonucleic acid molecules, microRNA and small interfering RNA, are central to components to the RNA pathway. Once mRNA is degraded, post-transcriptional silencing occurs as protein translation is prevented. Transcription can be inhibited via the pre-transcriptional silencing mechanism of RNA, through which an enzyme complex catalyzes DNA methylation at genomic positions complementary to complex RNA or miRNA. RNA has an important role in defending cells against parasitic nucleotide sequences and also influences development of organisms. The RNA pathway is a naturally occurring process found in many eukaryotes and animal cells. It is initiated by the enzyme dicer, which cleaves long double-stranded RNA molecules into short double-stranded fragments of approximately 21 to 23 nucleotide RNAs. Each RNA is unwound into two single-stranded RNAs, the passenger strand and the guide strand. The passenger strand is then cleaved by the protein argonaute 2. The passenger strand is degraded and the guide strand is incorporated into the RNA-induced silencing complex. The RISC assembly then binds and degrades the target mRNA. Specifically, this is accomplished when the guide strand pairs with a complementary sequence in the mRNA molecule and induces cleavage by AGO2, a catalytic component of the RISC. In some organisms, this process spreads systemically, despite the initially limited molar concentrations of RNA. RNA is a valuable research tool, both in cell culture and in living organisms, because synthetic RNA introduced into cells can selectively and robustly induce suppression of specific genes of interest. RNA may be used for large-scale screens that systematically shut down each gene in the cell, which can help to identify the components necessary for a particular cellular process or an event such as cell division.